So again, here's one granum, a stack of thylakoids, and here's one thylakoid. And we're going to zoom in on just one thylakoid membrane here. Don't be shocked. Okay. This looks very complicated, but we're going to go through it step by step. All of this is the light-dependent reaction, so the first step of photosynthesis. So what actually happens? Well, this is, first of all, just for clarification, this right here, this fat, long thingy, is the outer membrane of one thylakoid, okay? So this line is this line. Just the outside. Okay, we're gonna start here. Light hits our plant. And inside photosystem 2, the first step of our reaction, light waves at 680 nanometers are picked up and excites an electron. This excitement then, an electron is excited inside the photosystem 2, inside its reaction center. Okay. The now excited or charged electron moves on to a protein carrier inside the membrane of the thylakoid. Then it moves on once more, and here is an interesting thing. The excited electron then acts as energy for the active transport of protons across the thylakoid membrane. So, to say that in other words, these energized electron powers this protein pump so that a lot of hydrogen, or whatever you want to call them, protons, are pumped into the lumen of the thylakoid. Then step 3, the electron is pumped into another protein carrier, this time outside the membrane. And for each step it is pumped, it has lost some energy. So by the third step, it doesn't have a lot left. So after the third step, it enters photosystem 1. Here again, light, which I haven't drawn, but light hits the plant, and the electron is excited again inside of photosystem 1's reaction center. Photosystem 1 picks up light at 700 nanometers. Okay, so our electron is excited again. Then what happens is it moves on to another protein carrier, again outside the membrane. And then it moves here. This place I don't exactly have a label for, but we can call it the hydrogen reducer synthase or something. Basically, the electron powers the production of a reduced hydrogen acceptor. That means you have your hydrogen acceptor in an oxidized stage, so NADP, and you have two hydrogens from outside the thylakoid come together and create a reduced hydrogen acceptor. And this we need for the light-independent reactions later. Then the final step of the chain is this amazing phenomenon, what we've all been waiting for, the creation of ATP. The creation of ATP is known as phosphorylation, and it happens every time a hydrogen moves out of the lumen. Because before, a lot was just pumped in, and there was a negative gradient, or an unbalanced gradient inside the lumen of the thylakoid. But now, this pressure is equalized, you could say, and a hydrogen exits, and each time it does, ADP goes together with inorganic phosphate and forms ATP. Finally, it should be mentioned that inside the thylakoid, photolysis takes place. So you could say this is the only reaction that actually happens inside. All the other reactions here happen in the membrane. That's very important. So photolysis is the splitting of water molecules using light energy. So here we have a water molecule, and it's split to two hydrogens, and I guess a half of an oxygen. So for this equation to make balance, you'll need two water molecules. So already here in photosynthesis, the oxygen, the waste product, is actually created. And these hydrogens from the photolysis also add to this negative gradient, or this, what can you say, this overload. The negative gradient inside the thylakoid membrane. Okay, so all of this is the light-dependent reaction of photosynthesis. And then what happens is that our reduced hydrogen acceptor and our ATP goes into the cytoplasm of the chloroplast, the stroma, to perform the light-independent reactions.